Hey guys, this video is for educational purposes only. Please do not break the law.
Hi, welcome to Green Thumb Depot's MBA podcast. In this episode, we're going to be doing things a little differently. We have a special guest with us, and this week I'm going to be asking the questions for once. So, without any further ado, please welcome our special guest, Liam Spenser. He is the Director of Cultivation Solutions at AROYA, and today we're going to be talking about all things related to crop steering. Liam, how you doing? Welcome to the podcast.
Great, man. Thanks for having me. It's a pleasure to be here. Super happy to have a chance to sit down and chat with you and connect on cultivation stuff.
Absolutely. So before we get into it, would you mind just giving us a brief background of your history in this industry?
Yeah, so I started in the sort of commercial legal industry back in 2018 at Curaleaf. I joined the team over at their Florida facility, bringing that online. Was there for a couple of years, worked my way up to cultivation manager, and then moved to Colorado and took on the director of cultivation role for a company called Be Good. Ran 2 facilities for them in Colorado, about 300 lights ish total. So came from, several acres of greenhouse and a large scale build to some small craft indoor. And then from there, I started working for Scynce LED for a year as their cultivation, advisor, customer success person. And then about 2 years ago, I took on an account exec role at AROYA. And now I'm sort of here just overseeing our customer facing team and working day to day with all of our cultivators and growers, educating on irrigation, making sure they have all the right tools and resources that they need to get the success out of the systems that we deploy. So I have a bit of personal experience with the equipment. I ran a bunch of different systems and was really into controls and monitoring and all of that in all of the facilities that I've worked at. So kind of a natural transition into the technology assisted cultivation space.
All right. Yeah, fantastic. Definitely a lot of experience to draw upon in your current role. So, in a nutshell, now that you're at AROYA, what is a brief, let's say, elevator speech description of what you do at AROYA and what AROYA offers the industry?
Yeah, so AROYA is really just like a cultivation management platform that leverages instrumentation and sensors and precision sort of irrigation practices and a software that allows you to access and read that data in order to, drive the most optimal strategies to get the best outcomes in the plant.
Fantastic. Okay, so you guys have products, sensors, essentially, and controllers, and you also have software that manages it. So before we get into all of those details, for those folks who aren't familiar, can you give a brief description of what crop steering is and some of the factors that are involved?
Yeah, crop steering is really just sort of like a term used for precision cultivation methodology that leverages data and feedback to drive decision making on changes that you make within the plant. So that's irrigation strategy, environmental controls. We're really just managing plant responses and plant cues to trigger the plant to perform and behave and grow in ways that are sort of conducive for optimal production.
So would it be safe to say that anybody that's growing is doing crop steering, but some people just have no idea where they're steering their plants to go, and you try to help them get to the right destination, maybe?
Yeah, I mean, everybody's crop steering. It's how intentional are you about the practices and the things that you're implementing and the ways that you're responding. So like defoliation is crop steering, right? You're like creating a stimulus on the plant and triggering a plant response. Do you know exactly what it is and what's happening from a hormonal level? Maybe not, but is that just something that we've all always done since the dawn of cultivating cannabis? Yeah.
Okay, fair enough. Okay. So with AROYA, I know I'm very familiar with your water content sensors. What other types of sensors do you guys provide, or is it primarily the water content sensors and then you integrate other sensors?
I would say that our most popular and most commonly adopted generally is the Teros sensor. So originally the Teros 12 and then the new Teros 1 sensor. That sensor is specifically designed for substrate in cannabis. And then we also have climate sensors. We have, apogee light sensors. And then we also, our latest sensor creation is the drip and drain station, which measures drip irrigation volume, sort of like in your catch cup. And then the runoff sensor, which captures data, time series data in runoff from a plant, which you can get water volume, EC, and then a pH add-on.
Fantastic. So in that regard, with that runoff sensor, you can basically couple that with other sensors so you know what you're feeding your plant, what's going in, and then you know what's coming out. So you can catch any sort of issues that may be happening and you can really have a better idea of where your plant's going. Fantastic. So how much of crop steering is more related to irrigation and drybacks versus the environment, or would you say it's just basically everything is important, or are there some factors that are more important than others?
I mean, I think that there's a big misconception that like crop steering is entirely predicated on irrigation. So there are things that are within our control and things that we can respond acutely to steer the plant in the direction in a response. So for example, your environment, if you have limited controls ability over your environment, we're talking airflow, we're talking temperature, humidity, CO2, light intensity. Some of those can be fixed, fixed variables, I should say. So, let's say you can only set one lighting zone for the entire room. Now, what are the other factors that you can make changes and decisions based off of that sort of limiting factor, we would call it, right? So irrigation is probably the easiest like queue to send. It's the fastest response queue that we can send because we're constantly delivering irrigation events as opposed to like tweaking and tuning the light and the airflow and the temperature, those sort of things. However, the environment is the context in which the decisions that we make on the irrigation happen. So if you have an environment that's out of range, like the room's too hot and then the transpiration slows down, or the room's too cold and transpiration slows down and the plants aren't drinking as much, you would adjust your inputs accordingly. So your input EC, your input volume, the amount of shots, the window of your shots, those are things that we can acutely adjust to respond to limitations in our environment, so to speak. But the main crux of the input on the plant happens from the biggest driver is in photosynthesis is light intensity first. So you have, you got to think like, what is the photosynthetic equation and how are we backing our way into what we're doing to manipulate that?
Right, right. And as we've discussed in previous episodes, essentially all of these environmental conditions, they're all linked together. You can bump your watts up to 1500, but if you don't have the right VPD or you aren't feeding them the right amount of nutrients, the plant's not going to be able to handle that extra energy. So everything has to be dialed in and synchronized together. Would you agree with that statement? It sounds like you're saying.
100%. I think, there's a lot of different ways to conceptualize limiting factors. At the end of the day, the plant has a genetic ceiling. And in order to achieve that, it's just like the human body. Like if you work out and you don't eat enough, you won't gain weight. If you don't work out enough and you eat too much, you won't lose weight. The plants really behave similarly. So, the more light, the more CO2 you give it to a certain point, the more you have to increase the feeding and the irrigation, and those sort of things all have to follow.
Absolutely. Makes a ton of sense. So maybe let's just get into the irrigation component right now. And I know that's something you deal with, bread and butter every day. So, is there a particular water content range that you like to stay in? And maybe we can define a few terms for the folks like drybacks and, saturation and drainage and whatnot. And, does that range of saturation of your media, is that dependent upon the media as far as where the optimal range is, stuff like that?
Yeah, I think we have to look at it in the context of what media we're growing in, what our planting density might be, what our veg time might be, and then also, what our strategies are. So I typically say for like cocoa, it depends on the chip to pith ratio. And most cocos sit in that like 70-30 chip to pith range. So you'll see like somewhere around 65% as what we would consider field capacity. Field capacity is just a term that means the total amount of water that block can hold at full saturation before it achieves drain. And so most blocks can hold on to about 65 of that percent of that total volume of that block. So let's say you're in a 1 gallon pot, 3.8 liters, 65% is probably somewhere around like 2.3 to 2.2 liters in that block. And so that's how I like to frame it, is like, we're sitting at field capacity. That's a max water content. There is a slight difference between max water content and field capacity. Those things could change. But in a broad sense, to, simplify things, I like to achieve field capacity in whatever block I'm in every day. Some rock wool you might even see on our sensors up to 80%. Again, that's also, there are different sensors that use different tech to read different variables. Their calibrations could be different. So those are all sort of potentially, they could be variables. But I say, as a general rule of thumb, cocoa, you're sitting somewhere between 65 to 70 in a higher pith ratio, and maybe like 50 to 55 if you're running like a cocoa perlite blend, like a Mother Earth or something. There's still a lot of people out there that use perlite. I personally don't think it's necessary in a crop steering style, like precision irrigation methodology. Larger pots definitely make sense. And most of what we see is people running those like 1 to 2 gallon cocoa blocks, or a 6-inch rock wool block, or a slab, a triple slab. So yeah, in those, instances, highest water content you'll see is probably somewhere between 65 to 75.
Okay. And you like to hit that max capacity at least once a day. But then there's also, obviously there's a low end that you try to stay above, right? What are we looking at with the low end of water content?
Yeah, so on cocoa, I really don't like to lose more than half of the total available water in that block every day. So if your field capacity is 70%, I rarely would go below 35 unless absolutely, necessary. Like, let's say you can't get your EC up, maybe you have, plants that just are struggling in certain ways and you really needed to push them a bit harder. Like, you have stretchers, plants that stretch excessively, things like that, where you need to really get aggressive on the dryback. Certain cultivars can handle it and other ones can't. But as a general rule of thumb, I try to never go below 30% water content on anything. Rock wool could handle it, but it may not be able to come back in terms of regaining that field capacity. You could get channeling, and the pore space could open up, and that lattice could not come back together. So a typical rule of thumb for me for rock wool just depends on the phase that you're in. Like, if you're in veg, so typically you don't want to be drying back too hard in rock wool and veg, like maybe 10-15%. And I very rarely go below 45% water content in rock wool until I'm getting towards the end and I'm really pushing generative steering. So, we can talk a little bit about what generative and vegetative steering are, but towards the end of flower, what we consider the ripening phase, where we're really pushing the plants hard to finish, that's when I'd be potentially dipping into the 30s in rock wool. And I think there's a fundamental difference between, what rock wool and cocoa can handle in terms of minimum water contents. And we can get into that in a little bit.
Okay. Well, yeah, let's talk about the phases: veg versus flower, early flower, late flower. I know from my own personal experience, even though I've been growing cannabis for a while, I was growing some peppers in the yard one time, and I had a nice garden. I would just love going out there and watering the garden, and my peppers never matured. They stayed green because I would go out there every day and water, because it was bringing me pleasure to water them. And then I got distracted. I didn't water them for a couple of weeks, and the plant itself didn't look too great, but all the peppers turned red. They were ready to harvest. So, there's a similar thing going on there with cannabis. But yeah, why don't you walk us through, let's start with veg. What are we looking at as far as the numbers, and what are we trying to do with crop steering in the vegetative phase for cannabis?
I kind of want to stay away from hitting these very specific numbers. I mean, we can talk in general ranges, but I think that it gets a little misconstrued in terms of what we're really trying to accomplish. So I'd like to break it down from phases and stimulus. So we have the vegetative phase of growth, which is the 18 plus hours, anything over 12 hours really of light, typically that 18 to 24. And then you have your generative, or your flowering phase. And flowering can be broken down even further. Some people break it down into 4 phases. For simplicity's sake, conceptually, we break it down into 3. So we talk about the stretch phase, which is the first 3 to 4 weeks of flower, depending on the cultivar. We have the bulking phase, which is what we would consider a more vegetative irrigation phase in flower. And that's sort of weeks 4 to 6, maybe 7, depending on the variety. And then we have the finishing, the ripening phase. And that's, again, more of a generative irrigation strategy. And so we have those three phases in flower, and then we have vegetative and generative steering. So those are just stimulus on the plant, right? So like vegetative stimulus is trying to enable the plant to have as much access to resources, as much abundance, make life as easy, as simple as possible for this plant to have access to everything it needs at very little stress. And I like to stay away from the term stress in production because it can be misconstrued into like stress can be a bad thing, right? Like we don't want to stress the plants too much. We want to stimulate them. And so, talking into that generative style of irrigating or cultivating, you're pushing the plant a little harder, right? Like you're telling the plant, hey, I want you to reproduce. I want you might die soon. This is not going to be easy for you. So put on all of the material necessary for you to have the best chance of success of reproduction. So building more bud sites and flower nodules, building more nodes, building less leaf tissue, less stems, internode spacing gets smaller. Maybe you have lower leaf area index. Once your leaves have been produced, you're pushing the energy towards reproductive organs and root development, that sort of thing. So generative stimulus is more, we would call those bigger drybacks, right? Like deeper drybacks, higher substrate EC, more oxygen in the pore space. As you remove water, oxygen fills that pore space. So if you dry back harder, you have more oxygen. And so those are the things that we're playing with from a higher level.
Okay, fantastic. Yeah. A lot of what you were just talking about, we hit on at a very basic level. In last week's episode, we were talking about grow media and oxygen at the root zone and so on and so forth. So let me ask you this. How are beneficial microbes interacting with this crop steering? Do you find yourself recommending different microbes at different phases? Are they affected by these drybacks or mostly unaffected, or some more than others?
I think the science is still widely not fully understood on how what we're doing is really impacting the life cycle of the microbes, because the flower cycle is so short for the life cycle of reproductive cycle of things like mycorrhiza and fungi. Bacteria reproduce a little bit more quickly and adapt more quickly, but still sort of takes time to establish a colony and have them, really grow and respond. So again, back to the oxygen conversation, right? Like more oxygen availability, more potential for faster growth for aerobic bacteria. Fungi, still a slightly different mechanism there, but the more root zones, the more root sort of surface area that we have to cover, the more we can create those symbiotic relationships with the mycorrhiza, and they can attach to those root hairs and create that relationship. The problem that we have with this is that, when we're feeding a purely synthetic fertilizer routine, right, a lot of times those bacteria and the plant interactions aren't predicated on the sugars that are being produced to feed the microbes. And so you have to supplement, because you're basically providing direct chelated minerals to the plant root zone. You're just mainlining all of those macronutrients into the plant. The buffer zone for the mycorrhizae and the bacteria, they don't need to work so hard. They're not critical to the operations of that plant with those types of fertilizers, because typically in soil, those plants are breaking down humates and all these things to create nitrogens and phosphorus and potassium and deliver that directly to the root zone. So what we see a lot with people using microbes is what they'll do like a synganic is the terminology, or biosynthetic, or whatever you want to call it, where they're using fertilizer, but they're also putting in like fulvic acid and, other types of humates and amino acid chelates and other, sort of organic enzymes and things that build a biological ecosystem in the root zone that allows those organisms to interact with the roots and the sugar exudates that are coming off the roots in a more effective way to promote growth. But again, it really comes down to oxygen in a lot of cases. So steering the plant, we want to build and establish that colony early on in veg. So on those transitionary phases, like transplanting into your cocoa or transplanting into your substrate, right at that moment, you make your applications and then you continue to feed it. And I think sometimes there's an antagonistic relationship with high salinity environments in the root zone and certain species of microbes that don't operate well. So figuring out which microbes can survive and thrive best in those high salinity environments is really important too, because there's no buffer capacity. Cocoa has more buffering capacity due to the cation exchange in the root zone in the substrate. Rock wool, not so much. So there's less for those microbes and organisms to hold on to, and alkalinity is a little bit lower, so there's not a lot of buffering capacity. So that's something that we still don't fully understand, at least I certainly don't, and a lot of my colleagues we're still understanding how these things are specifically happening in those rhizospheres.
All right. No, those are some of my favorite topics there. We could probably have a whole podcast on rhizophagy and exudates and stuff like that. And for folks that don't know at home, maybe you can briefly explain the role of exudates with plants and, kind of how that's evolved with their relationship with bacteria, kind of like the natural way that it would happen, before we started doing everything that we're doing in CEA.
Yeah, I mean, root zone management is present in every crop and every type of plant and growth because the plants require nutrition somehow. So in the natural environment, the microorganisms were that sort of conduit to drive nutrients into the root zone of the plant. And so they were taking and breaking down these, rock phosphates and a lot of these like mineral dusts and things that were present in heavy sort of mineral rich soils and breaking those minerals down in a way that the plant could uptake and absorb them effectively. And so they co-evolved with plants. So you have like nitrogen fixing bacteria and other types of specialists in the root zone that allow in the rhizosphere that allow those plants to use and absorb those nutrients, right? So when we're directly injecting chelated and easily absorbable nutrients in the root zone, it sort of bypasses that system. But the reality is a lot of these organisms rely on the exudates of the plant, so the sugars that the plant puts out. So again, back to the photosynthesis equation, light plus CO2 plus water is sugar plus, oxygen, right? So plants are producing sugars as a result of photosynthesis, which feeds the microorganisms in the soil, and therefore they're able to expand and grow, and they take that oxygen and then they pull in those nutrients and break down the soil and deliver that back into the plant tissue.
Yeah. One of my most eye-opening experiences was really getting into that rhizophagy and understanding that the plant's actually attracting the bacteria to the root tips with that sugar-rich exudate, and then the bacteria will actually be sucked into the roots, and the bacterial wall will be dissolved, and all the ions sucked out, and then in some cases, not 100% of the time, that bacteria will be ejected through the root hairs back into the soil to go out and go harvest some more minerals for the plant, essentially. So it's almost like, it's an exchange, right? It's like these little bacteria out there living in the wild, collecting all these minerals and then coming back to the plant to exchange those minerals for sugar, almost like a form of currency. You're coming back to the city and exchanging your wares is how I explain it. But yeah, I think yeah, I think it's fascinating. And then, with regards to exudate, one thing that I always implemented in my grows was adding some sort of sugar to the nutrient solution. And I don't know how accurate this is, but my line of reasoning was, at least at the time, I was led to believe the plants can produce, I don't know, nearly 50% of their sugar production can be sent down to the root zone to attract microbes. And the plant can sense the sugar content in the rhizosphere or the root zone. It's the same thing, right? And when it's high sugars, then they know they don't need to exude as much sugar. And so they can send all those sugars north to the colas. So by augmenting with a cheap carbon source in our nutrient, that can actually attract the bacteria where it needs to be, keep the bacteria healthy, but allow the plant to focus its photosynthetic energy on cola production. Would you say that's an accurate statement, or are there some caveats I should update that line of reasoning with?
Yeah, I think that's probably pretty accurate. I wouldn't say that this is like my main topic of study and focus, but I would say that there's a huge relationship between plant health and sugar content in plant tissue. You know, Brix levels is what we've historically always calculated in terms of like what is a baseline for tomatoes and other crops. And cannabis is relatively similar, maybe 12 to 13% in organic production is what you typically see. You usually see a lot lower in purely synthetic production of cannabis. So there's a lot less of a buffer zone. You're kind of steering much more quickly and responsibly. A lot of things can derail more easily when you don't have that, high Brix buffering capacity for plant health. you can still grow extremely healthy plants with purely synthetic fertilizer. You just have to be much more precise and there's lower margin for error. So I think to your point of like, where does sugar allocation go in the plant? I think it's purely dependent on what that plant determines as what is optimal for its health. So if the plant tissue, where does it prioritize where it sends those sugars? Does it need it in the root zone or does it need it in the tissue? And where is it allocating that in terms of a limiting resource, which steers us back towards the conversation of, are we steering the plant generatively or vegetatively, and how does our stimulus and what are our inputs on the plant determining the plant's response and allocation of those sugars? So if we're trying to create a lot of scarcity in the resources of the plant, it may move its sugars differently than when there's full abundance of water and nutrition and humidity in the air and all those sort of things, right? And so that's something that, I think is widely not measured enough. Probably we certainly don't have instrumentation specifically to measure biology, sugar content, exudates, all those sort of things in vitro in real time. We're just using the measurements we can take as a proxy for plant health. So like we're taking the water content and the transpiration rates, how quickly it's moving water through the tissue and how that's impacting the humidity and all the other environmental factors to say, hey, this is a healthy baseline standard for this plant with this, stage in its growth cycle and that sort of thing.
Yeah, no, that makes sense. Now, you mentioned Brix earlier, and yeah, that's a huge topic there. Do you guys measure Brix with like a refractor meter or Brix meter? I forget the name. Or do you use that data in your analysis?
Not really. I mean, that's if we're gonna like kind of bring in some more comprehensive analysis on plant health, and that's sort of out of the scope of what AROYA is really, their main goal and purposes. That's sort of just like a contextual, like, are we feeding the optimal nutrition to this plant? Do we need to go do plant tissue analysis or sap analysis and test the plants in our customers' facilities or work with a partner, a lab that's going to test that because maybe they have something wrong that we can't figure out or explain. That's a little outside of the scope. But just from a principle perspective, it is important to know the context of some of these other data points to just have referential information on your cultivar's responses to the system that you have them in. And when you're talking like commercial cultivation, like you can't always adjust your feeds per cultivar. You can't adjust your nitrogen ratios just for Blue Dream or just for LCG. You may have to just average it out for all of your cultivars. So if you're testing, you may find one that has a different preferential uptake of that nutrient, and it may, as a result, have a lower Brix. So maybe that cultivar just doesn't perform well in your environment. You can't try to specifically cater to that, And so that is where extra data sometimes becomes cumbersome and noise and not so much actionable.
Sure. No, that makes sense. Data analysis paralysis potentially. Okay, so let's go back to the drybacks for just a moment. And one thing that I see a lot of folks doing nowadays is they're using these very short shots of irrigation and having a lot more irrigation events per day, but a much lower quantity per event, which in my mind is keeping that water content right in that sweet zone without going higher than you want or lower than you want. Is this something that you guys recommend over to, or is that not necessary, or there's some drawbacks to doing things like that?
I think, I think we can dive in a little bit on what we mean when we define irrigation events, and maybe that will shed some context into what we're talking about here, because a lot of times, people get caught up, lost in the sauce on like shot size, like being like, I want to give a very small or big shot. I want to pulse feed and send a bunch of shots. When we look at the big picture of our irrigation strategy, we have a couple key levers that we pull that really define our strategies, and we back our way into that based on what we're trying to accomplish. So that could be shot size, which would be volume or duration based on how you're irrigating. Usually a duration equates to a volume. So like a 1% shot on a 1 gallon pot is 38 mils. So how long does it take for you to run your emitters to get a 38 mil, shot? So usually if you have a 1-to-1 where you're doing 1 gallon per hour, or, ideally what you'd want to do is like size your emitters so that you have a 1% shot being like closer to 1 minute. So if you want to do a 5% shot, you do 5 minutes. If you want a 7% shot, which is about as big as I like to do, you got a 7-minute shot. Those are the kinds of things that I like to I usually suggest smaller the better on emitter size, but if you're going huge on your pots, like I typically don't recommend bigger than a 2-gallon pot. But like in a 2-gallon pot, 1 gallon per hour is fine. 2.5 emitters. Rockwool, smaller cocoa, I almost always recommend the 0.3s, 0.3 gallon per hour emitters, just because you can fine tune your shots and run longer shot sizes. Problem you'll run into there is you might have overlap if you have a lot of zones that are going in sequence as opposed to, if they're in series as opposed to going parallel, where you can have multiple shots fire at the same time. If you have like a certain type of irrigation timer that says this zone fires first and that's done, then this zone, now you have to wait 7 minutes. Now by the time you get to the last table in the room, it could be an hour after that first zone goes. So you have to be able to manage your constraints on your system. But usually we talk about shot size and shot interval and shot frequency. So that means like, size of shots, the time between shots, and the number of shots that you're running. And when we look at those, I usually like to back my way into those numbers by what my irrigation window needs to be, by what I want my transpiration to be before I start irrigating. So my last shot of the day before to my first shot the next day is my window of dryback, and my first irrigation to my last irrigation is my irrigation window. And so I'm managing my irrigation window to manage my dryback window, to manage my dryback percentage, and then stack my EC as a consequence of that. And so then I'm going, okay, well, let's say if I have a 2% per hour transpiration rate over the course of 24 hours, if I'm averaging 2% over 24 hours, that's a 48% dryback. If I have 70% field capacity, 48, if I didn't irrigate at all, by 24 hours later, I'm going to be down to, 22%. And so that's usually way too much. And so what I want to do is cut that down to maybe 35%. So how long do I need to irrigate? That's maybe 3 hours, 2 to 4 hours. So usually our irrigation windows are defined by hours. And then we go back our way into like bigger shots less frequently is more generative, smaller shots more frequently is vegetative. So like somewhere around a 3 to 4% shot is like a vegetative shot, and anywhere from like 5 to 8% is more generative. And so like you just do the math there and say, well, if I'm running 8% shots and I need to regen my block back to 70% from 35, well, 35 divided by 8, now I get my total number of shots, and then I can space it out in that window to hit that target dryback range. So now I can say, for example, 8 shots or 8% shots, I've got 5 of those shots. 8 times 5, that gives us 40%, right? So now we have a 40% irrigation volume per day. And we may not be drying back 40%. Maybe we're drying back 36 with 10% runoff, which gives us that other 4%. So we're managing a lot of variables and factors at once. So I don't like to say in absolutes like 1 or 2% shots. I like a range, and I like to manage my windows and back my way into those numbers based off of what I'm trying to do with my window, with my dryback window. So I think window is more important overall than shot size and frequency.
Okay. No, that makes sense. So you're basically determining the amount of volume that you want to irrigate in a particular day, and you're dividing that up over the course of the irrigation period during the day. And then if it's vegetative, you're going to do more frequent, smaller shots, and you're going to maintain a more even keel on your water content, not as much fluctuation up and down. Whereas in the flowering phase, and as you get closer to the end of flowering, you're actually having more fluctuations because you're doing longer irrigation, longer shots, and letting it dry back more before you hit it again. Would you say that's an accurate sort of overview?
Totally. And a lot of people like to break this up into phases. So you have like phase 1 irrigation is from peak dryback to field capacity plus runoff is your P1 phase 1 irrigation. And that could be, 4 shots, like I said, 4 to 5 shots at 6 to 8% on generative. And then you're going to do those every 30 minutes. Or if you're vegetative, you're going to do like 9 shots every 15 minutes at 3%, which is, 27%. Plus you're trying to hit a bunch of runoff. So maybe your dryback's 22%, and then 5 of that is all just straight drain to try to keep your EC down, right? So those are all the things. Then you have phase 2, which is your maintenance shots, which is where you're keeping the water content at close to your field capacity. You could be slightly sub field capacity. You could push runoff in your P2s. That just depends on what you want your EC to do. And that's kind of that's a metric we use to determine that. And so, the time between those shots, that window is just to manage your dryback. So let's say you've got a phase 2, let's say you want your total irrigation window to be like 8 hours, but you're able to achieve field capacity in the first 2 hours. Now you have 6 hours of window to play with on maintenance, where you could say, I want my inner shot dryback to be more generative, maybe 5%. I'm drying back at 2% per hour. Maybe I need a 2 hour and 15-minute interval between my P2s to get that 5% inner shot dryback, for example. So that's how we back our way into like kind of calculating what those are going to look like, is like, how often do I need to shoot water into the plant to regen that dryback that happened in between those shots based off of my transpiration rate per hour.
Okay, makes sense. So P1, P2, different uses for those. Your P1 is always your first irrigation of the day when you're trying to get everything where you want it to be. P2 is maintenance. Okay. And then, you mentioned stacking the EC. This is really important. People talk about this a lot. Can you just kind of give us a basic overview of what stacking the EC means?
Let's just kind of highlight what the purpose of EC in pour water EC in the substrate is, because that's what we're measuring. The point of pour water EC is to build osmotic pressure on the root system so that we can stimulate the plant to make it easier or harder for that plant to move water through the tissue, through the stomata, through the xylem tissue up to the stomata and the leaf tissue, whatnot. So, because you think of the plant as sort of like this straw, the longer the straw, the higher the EC, the more pressure on the root zone, the harder it is to suck that water. So in generative irrigation theory, when we're steering generatively, we want a higher EC in the context of what our VPD is on the plant, because that VPD is the suction force. That's the mouth that's pulling the water out of the leaf. So the higher the VPD, the higher the suction force. So if you bring the EC up too high, now you shut down the system because there's too much demand for how easy it is for that plant to move that water. So you've broken equilibrium. So we manage EC based off of the context of VPD and off of the phase that we're in. Generally, we push higher substrate ECs, pour water EC, during the stretch phase, during the generative phases of growth, so stretch phase and then ripening. And we try to drop that a little bit during bulk and during the veg. We try to avoid going too heavy on stack during veg, because we're trying to build a plant and build a root zone. And that high EC is going to really stimulate that plant to say, oh, I need to create reproductive tissue. So that's the point of stacking EC. What we're doing when we're stacking EC is we're drying the plant back and mitigating runoff so we can build an accumulation of salts in that substrate that builds that osmotic pressure. So there's available salts and unavailable salts. You have salts that have crystallized, become unavailable. Those can create pressure on the root zone. And you have the available salts, which the plant can use and take up. And when it doesn't need, it stays there and it just continues to build that pressure as that substrate dries back. The salts dry out, they crystallize. So like when you push more runoff, you're flushing essentially the media of those excess salts. When you push less runoff, you're accumulating more of those salts. And so you're building the pressure on the root zone. And so that's why we push high input ECs in the beginning, so that we can accumulate salts faster than the plant can really use them. So we can use that as a tool to leverage building EC and building generative stimulus on the plant. And then when we want to back off, we reduce that. So that's where we manage that with drainage. So the idea is to like stack EC in the right time to create that stimulus, and then adjust it and push more drain when we need to mitigate that.
Okay, that makes sense. So the higher the EC, the harder it is for the plant to move water up to the top. The higher the VPD, the easier it is for the plant to do that. So these are two different factors operating on different ends of the plant that are used in crop steering. So, and just as a recap for folks, so the VPD is when you have a high VPD, that means that the air, the atmosphere in your grow can absorb a lot more moisture than it currently has. So a high VPD creates a suction force, a sponge that's soaking water up. The plant is emitting water through transpiration, so it helps draw that through, like you said, the straw. I use the same analogy. Whereas on the other end, in the root zone, a high EC, all those salts are going to want to keep that water down in the root zone, and it's kind of like one's the accelerator, one's the brake, and you kind of have to match them to get the right throughput of moisture through the plant metabolically. Is that sort of, I don't know if that's the best analogy.
No, yeah, there's a lot of analogies we could use here. I love a lot of these analogies because they kind of talk to different aspects of the biological system. But, the suction force, analogy as a vacuum is a good one because, the main force acting on the plant is the environment. And the plant's response is modulating its water regulation, where it puts that water, how much gets out of the stomata, if the stomata is open or closed or not. So ideally, lower VPD is more of a vegetative stimulus on the plant, and higher VPD is more generative. So like a higher VPD could be more stressful for the plant because the plant now can't modulate its water transpiration rates on its own. It now has to close the stomata to stop water from leaving the plant, which means once it closes the stomata, it stops growth. And so when you have too high of a temperature at too low of a humidity, then your VPD is too high. Now it's pulling too much water out of the plant. So now the plant has to respond by protecting itself and self-regulating. So it could shut down. So the idea is to maintain an optimal VPD range for us to be able to crop steer through irrigation. Because if we try to do both at the same time, where we're trying to push high VPDs, we have to be very careful on how we manage the root zone. And then if we're trying to steer too much, we can't steer very easily in irrigation if our VPD is constantly fluctuating and your VPD graph looks like a roller coaster. Because now you have open and close and open and close, and you can't predict transpiration rates. So the idea for us is create as much predictability in the system, so for us to not be entirely responsive to the environment, to the plant, and be proactive and send stimulus and triggers and cue the plant, as opposed to just reacting to everything.
Makes a lot of sense. Absolutely. Yeah, there's definitely a sweet spot with VPD. You don't want too low, that's going to shut everything down, and you can't have too high as well. So within that range, though, there is some room for adjusting to get the desired effect. But that makes a lot of sense. Okay, well, let's get out of the technical for just a moment and just talk about more of the products that AROYA has as far as the sensors and the controllers and the services, and just a brief overview of what you guys have to offer.
Yeah, I think, AROYA's offering has been, pretty consistent over the years. We've built and developed a lot of cool new sensors and features and tools and all that. But realistically, the most important thing is that you have the substrate monitoring tools. So the Teros 1 sensor. We also have climate sensors because, again, understanding the relationship between your environment and your root zone and knowing the context of what's happening in the climate allows us to understand a lot more about what to do with the root zone. And then you can see changes happening. So we have the climate sensor. We can measure light intensity. We can measure substrate water content, EC, and temperature. And now we can also measure input volume in EC and pH and runoff volume in EC and pH. We have a sensor called the drip and drain station, which is exactly that. It's a drip cup and a drain cup. You put your emitters in one cup and you put your plant on the other, just the way you would do manual, fill cups and manual drain cups on your tables. You're just automating that process. So you can look at exactly which shot hits drain, look at exactly, how much drain you achieved per shot per day. You can see when you hit field capacity, see if you're channeling, and then understand your total irrigation volume you've delivered to that plant day in, day out, spread over the whole cycle. And you can also see exactly how much drain you've pushed. And then you can use that data to understand how your substrate EC responds in correlation to your runoff volume in EC, and then potentially your pH as another trailing indicator. So we have those. And, from a sensor perspective, that's kind of the gist of the main sensors that we offer. We used to offer an inline EC sensor. But yeah, so everything operates wirelessly. It's off of a self-healing mesh network. So every device connects to every other device through this mesh network, and they all connect to a gateway. So it's easy to just deploy sensors because you have a little, box we call our nose, which is a battery and a, transmitter that sends data back to our gateway. Gateway plugs into the internet, and then you have the software platform. So with the software platform, you've got historical data over time. You got your harvest groups that tells you what happened in every run for every strain. You can use that to correlate with whatever your outcomes are, like your yield data, qualitative data. And then the other thing is we also offer, we have a lot of hands-on support. We offer crop steering calls with people where they can review their data. We also can, we do a lot of troubleshooting. We have a big support team. We do a lot of customer facing engagement too, on-site visits occasionally, things like that, things of that nature. So AROYA is really sort of this like all-encompassing cultivation assistant company where we're here to just help empower and bring growers up to speed and hit industry standards and benchmarks. So our goal is to use what we've learned with all the growers we've worked with and all the industries and all the crops and cannabis, and help bring that to the industry to help everybody level up, right? So we want everyone hitting whatever numbers they want to hit with what their facility is capable of. We try to be as responsible as possible with the advice that we give everyone and be realistic with the expectations. But we've seen a lot. And so I think we have a lot to offer the general industry from what are good best practices.
Fantastic. And when you're talking about all the different sensors and whatnot, these can be integrated into other systems as well, right? Like what's the languages that you guys use in order to integrate?
So I think a big misunderstanding is you can't just buy AROYA sensors and plug them directly into another control system. The idea is you take the AROYA software and you use a software integration like Modbus. We have Modbus protocols that can connect to other control systems like Rhythm and CCMS. We have a few that we've worked with. We're working on a lot of other ones right now. But that way you can just either pull in protocols to control your controllers on AROYA on your AROYA dashboard, or you can take your AROYA dashboard data, send it over to your control system, and then be able to see what's happening on your controller. So it's modular functionality, very kind of dependent on what your setup is.
All right. And now, at the risk of embarrassing you, I have heard and I've seen some data that indicate that your guys' water content sensors are essentially just, there's not really an equal out there, that you guys just really have the best sensors. Is that something that you would agree with? And is there a reasoning behind that? Is there something special in the technology that you guys have that others don't?
Yes, I would argue that I have really hardly seen anything out there that comes close on accuracy. There's a couple other companies that use the same technology as what we used for the Teros 12, which used to be the industry standard. So that was what we pulled from Meter Group. And the Teros 12 was a capacitance-based sensor. There's tons of other capacitance-based sensors out there. A lot of it has to do with programming and calibration and these other sort of factors. But the Teros 1 is a complex dielectric technology. So it individualizes the readings from water content in EC. The capacitance sensor uses an algorithm to measure water content in EC sort of off of the same signal that it's sent out, whereas the complex dielectric is sending two different signals to the root zone to pick up on water in EC. And so that's what sort of makes it like the most accurate and most responsive sensor. And it is sort of the newest and latest and greatest technology. It took us over a decade to develop something specific for high EC container-based cultivation using that type of media, whether that's specifically for cannabis. It's been applied to other plants, but cannabis is the only plant that we really steer with high ECs. A lot of problems that you would see with the Teros 12 is as the water content dips down below 30%, you start to see that EC spike, like sort of in a way that's not accurately representing what's happening in the substrate, and it becomes less useful information. And so we fixed that problem. So yeah, that's kind of like the shiny metal object part of the Teros sensor.
All right, fantastic. Well, thank you for that explanation. So, yeah, so essentially, it sounds like a lot of your work is very custom based. You got to get in, I mean, not necessarily the products, but as far as getting the right solution for the customers, you got to, get in front of the customer, understand what their needs are, what their media is, what their goals are, their cultivation strategy as a whole, what they're using their product for, are they doing extraction? Are they, just doing flower? Really understanding their entire business so that you can give them the best, absolute best advice that you possibly can. And I know you're extremely busy. So, any of our customers, if they want to get a hold of you, the easiest thing to do is give us a call, and then we'll review the needs and we'll set up a call with you or one of your employees and we'll get them on the right track. And yeah, we really appreciate having you here today, Liam. I know it's a lot of time out of your schedule, and boy, lots of good stuff to learn today, and I appreciate you for that. Any last words you'd like to let all our customers know and folks watching this video?
No, it's really exciting to get a chance to talk to you on this format and kind of get this sort of, exchange of valuable information. This is really what the industry needs, is more stuff like this, more people talking openly about, we all have different knowledge sets and different backgrounds and different strengths. And I think, just coming together and putting our heads together and presenting that to the industry really helps everybody level up. So I appreciate you setting this up. I appreciate you bringing me on, hearing what I have to say, and vice versa. It's been a great conversation. Like you said, anyone who wants to kind of get interested in AROYA through you guys, reach out to you first and we'll go from there. So excited to be working with you guys more closely, and hopefully we can do this again soon.
Oh yeah, we're definitely going to have you back. I can smell at least 3 or 4 other episodes that we could go with this. Maybe get a few other people on at the same time and have a little debate. Yeah, I'm a big proponent of being humble in this industry. I think as long as you maintain a humble mindset, that keeps you in a state where you're always willing to learn something more. You're not blocked off by your own ideas of what is and what reality is and what's right and what's wrong. And I'm always finding stuff out that some of the things that I learned, 20, 30 years ago, they're not really accurate today, and so you got to keep up with with the exchange of information, get it where you can from podcasts, books, whatnot, but stay on top of it and continue to learn, no matter what it is, but especially in this space. So very much appreciate you being on here, Liam. And with that, we'll conclude this episode of the NBA Growers Podcast, and focusing on crop steering and our special guest, Liam Spenser from AROYA. And we'll see you next week. Take care.